Okay, guys, so these are some DNA notes that I usually do on the document camera during class, but since that's not really an option, I'm just going to kind of do a narrated fill in the blanks note sheet here. You'll need these notes for the practice questions that follow up. You'll also need to scan them and show that you filled them in and turn them in online. And it'll help you with your study guide and your eventual test on this material. So I'm just going to kind of talk through it, and I will type in the blanks, the words as I go through. And if you print off this document, you'll just write them in as we go along. So if you guys remember, DNA is a type of macromolecule. We had 4 macromolecules: carbs, lipids, proteins, and the category that DNA falls in, which is nucleic acid. So DNA is a type of nucleic acid, and all macromolecules are composed of smaller molecules. Remember, those are called monomers. The monomer of a nucleic acid is a nucleotide. So a nucleotide, sorry, I can't spell or type. Okay, so a nucleotide has 3 parts. A nucleotide has a sugar. In DNA, the sugar is deoxyribose. In RNA, the sugar is ribose. It has a phosphate. And it has a nitrogen base. If you guys remember from learning about DNA in the past, the nitrogen bases are abbreviated A, T, C, and G. And those are able to actually hold the DNA strand together. So we have the 3 parts of a nucleotide. And remember that DNA is located in the nucleus. And it replicates or copies itself during the S phase of interphase in the cell cycle. So we learned about this when we talked about mitosis and the cell cycle. Interphase had G1, S, and G2 phase. Remember, the G phases were growth phases, and the S phase was when the DNA was replicated so that the new cell had a complete set of DNA. And so DNA is able to be made during this portion of the cell cycle, and it's going to rely on the rules of base pairing in order for DNA replication to occur. We recently discovered DNA. For the most part, it was the shape of DNA was found in the 1950s, which really isn't that long ago. They actually discovered that DNA was the genetic material in the 1800s, so that was a long time ago. But we didn't really know what it looked like until the 50s, when scientists started trying to analyze the structure. So James Watson and Francis Crick are credited, not edited. They're credited with discovering the shape of DNA. So they won the Nobel Prize in the 50s for determining the shape of DNA, and a lot of times we'll hear the term Watson and Crick. But someone kind of a lesser known or an unsung hero of DNA shape is someone called Rosalind Franklin. She was a female scientist, and in the 50s, they didn't really get a lot of credit. And she was working with someone called Maurice Wilkins. He was her partner, and they didn't really like each other. She took this picture of DNA over here using X-ray crystallography and shared it with her partner, who she was working with. And again, they just didn't really like each other. He didn't really respect her as a woman, and he really just wanted her to like get him coffee and stuff. So he shared this information with James Watson without her knowledge, and that was kind of the key piece of the puzzle that they needed to make their model work, so they could determine that DNA was actually a spiral shape, because they didn't know that. So now she's gotten more credit as we've learned that there's actually more to the picture. Hold on 1 second. Ansley's crying. I'll be right back. Okay, sorry. I can usually pause, but I don't know where the pause button is. So thanks for waiting. Okay, so we call this photo over here photo 51, and that was the photo that determined that DNA is actually a double helix. They could tell that by looking at this. And Watson and Crick were then able to use the research that they had along with this information to put together a full-scale model. So as I said, Watson and Crick base their model off of this picture so that they could actually come up with their Nobel Prize winning model of DNA. And later on, we did figure out that Franklin and Wilkins contributed. Again, Wilkins was a little sneaky about it. And she would have won a Nobel Prize. Maurice Wilkins did win the Nobel Prize with Watson and Crick. Rosalind Franklin would have also won the Nobel Prize, but she died. She also, some of her other research was involved radiation, and she died from cancer. So she was not able to receive the Nobel Prize because you cannot receive a Nobel Prize if you are not alive. Another person that contributed to the discovery of DNA shape, I do not believe he won a Nobel Prize, is Erwin Chargaff. Sorry, there you go. He contributed to the discovery of DNA shape by discovering the base pairing rule. So he analyzed the content of DNA and found that the percentage of adenine was always the same as the percentage of thymine. And so he figured that they must bond together and that the percentage of cytosine was always the same as the percentage of guanine. And so he was the one who taught us how the bases pair by analyzing the content. So putting all those pieces together, they were able to determine the structure of DNA. All right, so here we have this piece that is circled that is similar to what you labeled up here. And remember, that was called a nucleotide. So this is a nucleotide. And we can see here that DNA is made up of repeating nucleotides. That's why we say that is the monomer that makes up this long macromolecule. We can see here in our color code that adenine always pairs with thymine, that orange and yellow are always paired together, and cytosine always pairs with guanine, and purple and green are always together. The sides of the DNA ladder are called a sugar phosphate backbone. And then that's what this is pointing to. So we have the nucleotides. This is the sugar phosphate backbone, and then the whole molecule is going to twist, and we call this full twisted molecule the double helix. Also, these are anti-parallel. If you notice, all of these pentagons are kind of right side up, how you'd expect to see a pentagon, and then all of these are upside down. We call that anti-parallel, how they're both made of nucleotides, but these are right side up and these are upside down, and they're just going in opposite directions. So think about how, like, if you're driving on a road, you can have a two-lane road. Some cars are going like east, and the other ones are going west. So they're both driving forward, but they're driving in opposite directions. And so a two-lane road would be anti-parallel. They're driving next to each other, but one's going one direction, east, and the other one's going in the opposite direction, west, or north and south, whichever you prefer. All right, so as we mentioned here, when we look at this twisted ladder, we would consider this the ladder. These are the sides of the ladder and the steps of the ladder. Our shape that we coil into is called a double helix. There's not how you spell double. The DNA structure is a double helix. The sides are made of sugar and phosphate. And in DNA, it's a deoxyribose sugar and phosphate. And we call this the sugar phosphate backbone. So not super original there because it's made of sugar and phosphate. So we call it the sugar phosphate backbone. The sides of the DNA molecule run anti-parallel. That's what I was showing you above. I cannot spell parallel apparently. Meaning they run in opposite directions. So this is what I was talking about up here, where they are running in opposite directions. So the sugar phosphate backbone here is zoomed in so you can actually see the sugar and the phosphate. Here it's simplified, it's just like a line. And you can see that these are right side up, these are upside down, and we call that anti-parallel. Parallel meaning they're running side by side, anti meaning in opposite directions. Now these directions have names. It has to do with the number of the carbon that they're on. It has a lot to do with organic chemistry and how we number carbons. I wouldn't concern yourself too much with this, but it is important to know these terminology So we're going to go ahead and make sure we cover it. We say one strand runs in the 5' to 3'. The little apostrophe is called prime, so the 5' to 3'. This one that's right side up is going 5' to 3'. So up here, this is our 5' carbon, this one next to the phosphate. And then it goes to the 3' carbon, which is this bottom one. And then here it starts with the bottom one, so it goes 3', and then it ends with the top one next to the phosphate, so it goes to the 5'. Again, I wouldn't get too caught up in this terminology because it has a lot to do with organic nomenclature that you will learn later and the more in depth you get into chemistry and biology. But it is important to know that the anti-parallel means one strand runs 5' to 3', and the other strand runs 3' to 5'. Again, if you want to know, the one that looks like the pentagons are kind of right side up is 5' to 3', and the ones with upside down pentagons are the 3' to 5'. Our steps of the DNA ladder are these nitrogen bases. And they are held together, they bond together to hold this whole double helix together by hydrogen bonds. Hydrogen bonds are between complementary bases, and that means that only certain bases will pair together and they will be held together by hydrogen bonds. So in your DNA reading, you should have read about this, about how the hydrogen bonds are different between the two different bases. We can see here that the A and T only have 2 hydrogen bonds, and the G and C have 3 hydrogen bonds. So that'll be important when we talk about replication, because when they go to replicate DNA, we actually unwind the molecule and open it so that we can add new bases. And we usually open it in sections that have a lot of A's and T's because it's easier to break 2 bonds than it is to break 3 bonds. So the names of the bases are adenine, C is for cytosine, G is for guanine, and T is for thymine. Adenine, cytosine, guanine, and thymine. And they pair up so that A always pairs with T and C always pairs with G. So in your video, I think their memory hook is that apples in the trees and cars in the garage. So adenine will only pair with thymine, and cytosine will only pair with guanine. And again, A and T, apples and trees, C and G, cars in the garage. So use the rules of base pairing to determine the complementary base. So I also have these numbers here because I want you to remember the anti-parallel feature of DNA. So if this is 5' to 3', then that must mean this strand starts with 3' and ends in 5'. So now we can just do our rules of base pairing. So if I have an A here, A would always pair with T, T would always pair with A, T again with A, C, cars in the garage, pairs with G, T, C, A, T, C, G, T, A, and then G, G, T, A. So that's pretty easy. All you have to remember is A's and T's and C's and G's. Whenever you see one, you put the other. And here we want to find the base pairing error. We call a base pairing error a mutation. So we're looking for here a mutation that occurs where the wrong bases have paired together. So sometimes when we're making new DNA, they make a slight error where they accidentally pair the wrong bases together, and that can lead to problems later on. It could cause the cell cycle to malfunction and a cell to become cancerous, or it could be no big deal. So it just depends on how big of a problem the mutation is. But let's look here and look for problems. So good, good. Oops, so that's wrong. Make it red. Good, 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 wrong. Good, 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 wrong. Good, good, good. So there are 3 mutations there where the wrong bases are paired up. This is a little thing, DNA math, so you can kind of analyze the content of DNA. If a DNA strand is 18% adenine, predict the relative percentages of the other bases. So, okay, if 18% of the molecule is adenine, then we know that it's going to equally pair with thymine. So there's also 18% thymine. 18 plus 18 gives us 36. And we know that if we're taking a percentage of something, the total percentage has to be 100%. So if we've used up 36% and we have 100% as our total that we need to add up to, we can go ahead and fill this in right here. We have 18% adenine, 18% thymine. Again, that gives us 36. 100 minus 36 is 64. So 100 minus 36 is 64. This means the remaining percent that needs to be divided equally between the two remaining bases. So I have 64% left that I need to account for, but half of that has to be cytosine and half of that has to be guanine. So 64 divided by 2 gives me 32. And so the remaining percentage of cytosine and guanine is 32. And if you ever want to check your math, all you have to do is add up these four numbers and see if they hit 100. So 18 plus 18 is 36. 32 plus 32 is 64. And 36 plus 64 should give us 100. So that's how you know you did it correctly. Also make sure that A always equals T and C always equals G, and that you don't put them on the wrong lines. So here are some practice questions that are just a follow-up to everything that we just talked about. All of the answers should be above, and then you have the math questions right here that you will do exactly like we did right here. So first you find which base it would pair with, and then you make sure that those are the same numbers. You subtract that number from 100 and you get your remaining percentage, and then you divide that by 2 and put that next to the other bases. And then here's a diagram. I just want you to label deoxyribose sugar, phosphate, adenine, thymine, cytosine, guanine, and just circle what would represent at least one nitrogen base. So remember that you might not have guanine listed here, but what would guanine pair with will let you determine which one should be labeled guanine. And then the sugar and the phosphate will be found in our backbone. You could even identify which is the 5' to 3' and which one's the 3' to 5'. So these are the questions that you'll need to answer and scan along with the notes to prove that you finished it, and then just upload to me. Thank you. Bye-bye.